Yes, thank you.
Okay. Anybody have any burning questions we need to deal with first?
I have one topic.
Uh-huh.
What's mitochondrial and chloroplast DNA?
Do you want to discuss that?
Yeah, let's
Shall we go over it again?
Well, I don't want people they want to do something else.
No, okay. So mitochondrial and chloroplast DNA, let's just consider them completely identical, shall we?
Sure.
Except for that we don't have chloroplast in our cells, right? Because why?
Because we're not
plants. We're not photosynthetic. So basically, I'm sorry, when we're having this very intense conversation, can you keep your water bottles to a minimum? Thank you. Okay.
She's great.
So as we talk about these, just know mitochondria, chloroplast, doesn't matter which one, the same rules apply. Okay, here are the rules. There's only one chromosome in them. They only have one. How many do we have?
We have 2.
We have a lot.
23.
We have 23 pairs. So how many is that? Math geniuses?
46.
We have 46. We have 46 chromosomes, right? And they have, how many did I say? I forgot. One, okay? It's circular. It's a little singular chromosome. And it comes from which parent? The mom. The mom. Sometimes in plants they'll call that the ovule parent, O-V-U-L-E. The ovule, it's hard to say, parent, just because they don't really call plants mom and dad. So ovule plant means the egg, ova, egg, anything with the O-V-A beginning. So sometimes you'll see it referred to as the ovule, and of course the pollen is sperm. So if you see a pollen and an ovule, you'll know the difference. Yep.
So for inheritance, what do we call that? It's not sex-linked, it's
maternal.
Maternal.
Maternal. You just say straight up, it is maternal inheritance. Comes right from the mama. Okay.
That kind of just man, maternal. No.
No.
That's
No.
I mean,
Or
Paternal. Paternal because paternal means father.
It's
mom, maternal.
Oh gosh.
We're good.
Yeah. Mama and papa. Go with that,
I was
okay?
gonna say papa, but it made me uncomfortable, so I kept it.
Okay. So that's basically those two. Any questions about those two beyond that? Does that satisfy your
That's
curiosity?
You literally said, she said all that yesterday.
That's okay. Baylor needed a recap. It's fine. Anybody else have a burning question you want me to go with at the beginning? Before I just start popping topics up here and then you can decide what to do with them?
Can we just say skip if we don't want that one?
Yes.
Okay.
Although you have to agree. This is a democracy, not a dictatorship. Okay. A, B, C, D. Identify.
Oh, Lord have mercy.
Just try to do it without saying it out loud first. See if you can do it first because I want people to have time to think.
Wait, what are we doing?
You're telling me what is what? What are they?
Is there an interbank?
These are monomers of our big. Don't say them out loud. Because we're giving people time to
I
think.
know what A is.
A, B, C, and D. Commit to something here.
That's cheating.
Shh. No cheating.
That's upward.
Okay.
You think you know? Oh, Lydia's going a solid no. She doesn't.
I'm bad.
Lydia, can you narrow them down to like 2?
No.
No, dang it. Do you know one of them, Lydia? Just one.
No.
Ah, okay. Fine.
I think I know one.
Let's start with A. What is A? Does anyone know? Yeah, Lena.
Protein.
It is an amino acid. Amino acid make up what? What do they make up?
R groups.
Protein. All right, stop it, children. This is out of control.
I said amino acids, but I raised it.
You guys are just yelling out words. You guys are yelling out biology words like cuss words. Hang on, I gotta get the screen a little closer. There we go. This is gonna be the worst filmed video ever. Sorry, people that are watching this video. It's terrible.
They looked at it like I was stupid.
This is
Sorry.
protein.
Okay, so this is an amino acid. You should be able to recognize it. This makes up proteins. Okay? Oh, good Lord. Give me an example of an amino acid. Give me a name of one.
An enzyme.
Nope. Give me a name of a specific amino acid. I want you to trigger your brain way back in there. Yes. What? Ooh, close. That's a nucleic. That's a new.
Oh.
That's a nitrogenous base. It's on that chart. Do you know any of them?
I can remember the little things.
Okay,
that
what's
go-
one of them?
No.
I can only remember what they code for. I can't remember the name.
Oh, wait.
What they
no,
code
I-
for?
I got it.
Like you have your chart, you have your.
AUG codes for
That's
Here.
a start code.
yes, and do you remember?
Tyrosine.
Well, there's one of them, but yeah, methionine, mets. You know, we talked about that one a lot. Lysine. Okay. Yeah. So, there's something about me that I can't teach with the door open. I don't know why. I'm gonna leave it cracked in case someone comes along with review.
It's like the chart, right?
Something
Like,
about
on
the
the chart.
Yeah. So these are amino acids. So this is an amino acid, the codon chart, the one where you say AUG. Oh, that stands for methionine, right?
Yeah.
Methionine is the amino acid, that thing, right? And then it asks you, where's the R group here?
It's CH3.
Could you find the R group if they flipped it upside down?
Yes.
How would you find it?
CH3.
Well, it doesn't have to be CH3. It could be a lot of different things. Right. It's the opposite of this. It's the opposite of this hydrogen, right? So you look for your carboxyl, your amino group, and it's the opposite of that. Because I have seen one time where they put the R group on the top, and I'm like, what the heck are you doing? Yeah.
Do we have to memorize the chart?
Oh no.
If we need to
Oh, no. If you'd had to memorize that, I'd have told you that a long time ago. No, you do not have to memorize the charts. You need to be able to recognize an amino acid. Okay.
But we don't have to know which.
No, I don't even know which amino acid that is. I don't care because I always have a reference book, so I never have to look it up. But I do know the names of a few amino acids. Why I asked you that was because I wanted you to connect this to that codon chart. Like that's where I find amino acids is on that codon chart. So when it says meth, GLY, GLU, LYS, THR, all those are amino acids.
Wait, so
That's what they
codons are amino acids?
Codons code for amino acids.
Oh.
Your codon is like AUG. Like whenever you're using the code, it's like AUG codes for methionine. So AUG is the codon, and the amino acid is methionine.
And
Yep.
nucleotides like turn into like they get mRNA
Nuc-
transcripted and then
Uh-huh.
after that.
We'll
they
get there.
turn into the-
We'll get there. Yeah. Yep. A codon is the one that's on the mRNA. The anticodon is the one that's on a tRNA that matches up with it. It's the opposite of it.
Well, then what do they do once they match up?
Then the tRNA will leave that amino acid there because it's the correct one.
It's a double P.
So it's basically like here comes the tRNA, right? Here's the mRNA. tRNA goes, oh, my anticodon matches that codon. This is the right one. And it's transferring the amino acid to the ribosome, so it leaves it there. And then it goes.
So there's an amino acid attached
To
to the
the tRNA.
RNA.
Mhm. And we'll have a picture of it in a little bit, but yes.
The codon attaches to the anticodon.
Mhm.
And the amino acid joins the chain.
Mhm.
Okay.
Okay, cool. What's B?
And your nucleotide.
B is a nucleotide. Nucleic acid is what it makes. I just wrote the wrong word. It is a nucleotide. It makes up a nucleic acid. Spoiler alert. Give me an example of a nucleic acid. Those are not nucleic acids. Those are nitrogenous bases.
Oh.
What's a nucleic acid? DNA. DNA, RNA. And that's okay. You just answered too fast. Slow it down. Slow it down. Okay. Is this a purine or a pyrimidine? It's a purine. How'd you know that? Double ring. Right. That's good. That's good. This is a purine because it has 2 rings.
Oh.
If it only had 1 ring, it would be a pyrimidine.
So guanine or adenine?
Oh, is it guanine or adenine? I don't know. It doesn't have enough detail if I did know the difference. Yep.
Are there rings on all of them, or can we just
A-
ring nucleic acid?
A, C, T, and G, if they have it drawn out like this, they'll be rings.
I mean,
But
if we can't decide which
Oh.
for like
like
macromolecule it is.
that. Is that gonna confuse you?
No, because that one's just different.
Yeah. This is what it looks like. I mean, it'll always look like this. It'll either have 1 ring or 2. It's the only difference between them. So this is a nucleotide, by the way. It says nucleic acid, but it's polymer. Sorry. Somebody wants in. I should deny entry. Look, Amy, now you have a friend. Welcome, Stephen. Okay. Anybody know what C is?
R.
That one is actually a fatty acid, a lipid, yes. Is it saturated or unsaturated?
It's
Unsaturated.
Unsaturated has a bend.
Well,
Saturated is nice and smooth, okay?
saturated straight.
I don't know that you really care about that though. I mean, yes, it's in your standards, but it's, huh?
Could you tell us the fatty acid again?
So fatty acids have a few, they have a lot of carbons, a lot of hydrogens, but they hardly have any oxygen. So see that one oxygen there? Remember CHO? And we said that sugar is 1 to 2 to 1. 1 carbon to 2 hydrogens to 1 oxygen is the ratio. But then we said for fatty acids, there's hardly any oxygen at all. Like tons of carbon, tons of hydrogen, barely any. Okay? So last one then, that's a monosaccharide, which is it's carbohydrate. It makes up a bunch of polysaccharides. This is a sugar, right? Okay? I don't have anything to say about it. Just a sugar.
Do they ask us a lot about these? Okay, that's
No, they usually ask you 1 or 2 questions about recognizing, or maybe it'll be in there's not even always a diagram of them. I just would hate for you to, every once in a while there is, but for a lot of times they'll just ask you about, what makes up what. And actually it's more like, they'll have like amino acid or something in the question, and then you'll need to know they're talking about proteins. It's like you have to put it together. Like what is what? All right. Which one is a lipid? One's a sugar and one's a lipid. Yay! See the difference? There's only like 1 oxygen and the other one has like a bunch of oxygen. Do you see that?
Yep.
Okay. Because sugars don't have to be in a circle. It can be linear too. So you kind of look at that. I'm going to leave biochemistry behind because I don't like it anyway. Oh wait, there was this one. What one was unsaturated? Baylor.
Which
one's
one's
that?
B? Because it's not straight.
See, and do you see that double bond there in the middle? You see that double bond?
Yeah.
That's because it's not saturated with hydrogens. It had to bond to carbon again instead. Oh, what's going on here?
Oh, they're loving each other.
Oh, my heart.
They're making life.
Dehydration synthesis.
Yeah.
Yes,
you
I
guys
got
are
that.
the best. Dehydration synthesis. You guys rock.
Because
Because you're synthesizing a polymer, right? You went from 2 that were separate that only had 1 each into 2 together. So you synthesized them, you put them together.
And it's the
And it's losing water. There it goes, that little green guy is leaving. So bye.
what's the one that's going from a monomer to polymer again? What's that called?
This one?
Yeah, after dehydration synthesis.
Hydrolysis.
Oh, no, but you said it wrong. Dehydration synthesis is going from monomer to a polymer.
And then
That's what you said.
polymer to monomer.
This
is
one.
called hydrolysis.
This one is hydrolysis. Do you see it?
Yeah.
Do you see how it used to be 2 together and now it only is 1?
So
Okay.
I thought you read this
water
backwards.
goes back.
It's so like weird because it's like dehydration, like, but really it's like.
Losing water.
But then like hydrolysis.
Hydro.
Making water when it moves.
I know why I got so. Yeah, y'all.
I also know what you mean.
Just if you see a question that's kind of alluding to this,
draw it out.
draw it but just sit there and think about it for a second. It's okay to take a second and just sit there and think, right? You don't have to like do everything fast. Sit there and think about it for a second.
I drew the little picture on the mock. I remembered where everything was
Good.
on the little
Oh,
circle.
fun.
Oh, well.
Which one is thymine?
Oh,
Oh, let me give you a hint. G and C are triple bonded.
What?
G and C, it's that little thing in the top right corner.
C and D.
So which ones are C and G?
D and C.
D and C. The bottom 2 are C and G. See the triple bonds?
oh yeah.
Okay. And so which one is thiamine?
Yay.
A pairs with T.
Good job. You guys are so smart. Hey, what is this, by the way? What macromolecule is this? Nucleic acid and specifically DNA, right? How did I know? It's a double helix, but just in case it was a double helix RNA, how else did I know?
There wasn't a
Right. It has thiamine. Good. Where can I find DNA in a cell?
nucleus.
Okay, that's a eukaryote, right? So where else in a eukaryote? Uh-huh. There are 3 for a eukaryote, right? You see it? You got your nucleus, that's the thing at the top that's all balled up like that. And then you have your chloroplast and your mitochondria. 3 places where you could potentially have DNA in a eukaryote. Now that would have to be a plant cell, right? Because obviously we're never gonna have a chloroplast in our cells. So we only have 2 places. Where is it in prokaryotes? Not ribosomes, it's in the nucleoid region, in that area where there is DNA, basically. Okay, so there it is. Oh, look. Tell me about prokaryotic DNA. Tell me a little bit something about it.
It's
Circular. How much do they have? They should have 1, right? Oh, wait, what are we talking about? Mitochondria? Yeah, same.
It's singular stranded.
No, don't say stranded. It's just a single chromosome. It's double-stranded, just like DNA. I mean, it's DNA. It's just just one of them. It's just a single one. No from mom and dad, just one.
So they just have one.
They're just like our mitochondria and our chloroplast, exactly the same. That's what whole endosymbiotic theory.
But mitochondria and chloroplast, they're double stranded.
Mitochondria, no. Oh, they're okay. Are you saying stranded? Yes, they're double.
Double helix.
They're double helix. Yes, they're just a single chromosome. So they have double helix, but they only have 1 of them. Not like us, we get 1 from mom and 1 from dad, a double helix from both. Uh-oh, I just confused Adeline. Adeline, talk to me about DNA after class. Okay. Signaling for a protein, yay, moving on. Yeah, done with that biochemistry. Kiss it goodbye. All right, what is that thingy-majigger called?
A ligand.
A ligand. Yes. Who knew that? I didn't mean just him. That he's the only one that knew it. I know he said it. Did you know it? I'm asking if you knew it, Brooklyn. Who all knew that? That was called a ligand. Okay. Okay, so these are the steps. First, it's the signal reception, and then what do we call the next step?
Transduction. No.
That's correct. Signal transduction, because we're transducing the signal throughout the cell. And then what do we call the last step?
Activation.
There's an R.
Response.
Response.
Reception.
Okay.
So the ligand
The ligand triggers yes.
triggers the reception.
Ligand triggers that reception. Now, I guess that the ligand actually brings, it starts the whole process, and the cell receives the message from the ligand. Okay? So here's an adorable little thing going on here. I don't know if you can see this, honestly. It's so small. Can anyone see what's written in that little red ball? Can you see those? Like, what is the ligand? Can you tell me what the ligand is?
The red ball.
The red ball. What is it?
Calcium.
Calcium. It's calcium for those of you who are too far back to see. It is calcium. And so then, can you tell me, like, what is with those little antennae things? Can you guys up here see what they are?
Phosphorylation.
Yes, they're being phosphorylated, right? Those are little P in a circle for those of you who can't see it. So yeah, that molecule there, that orange MEK, is being phosphorylated, which we said a lot of times phosphorylation is a big part of passing a signal.
Wait,
okay?
why is it turning from red to green?
It's not. It's passing the message from MEK to ERK.
Wait, so why does it do the little P thing?
So phosphorylation, putting a phosphate onto a molecule like it's probably a protein. Putting a phosphate on a protein or taking a phosphate off can change the shape of the protein. And when it changes the shape of the protein, then it changes its function, and it'll like pass on the message to the next one. So it might release something, or so it's almost like that what I talk about Rube Goldberg machine, right? Where it's like waiting for the next thing to come along. And so the phosphorylation of it is like, oh, well, if I add this to it, then it might change the shape, and then it like rolls a ball out, or something. So
it's
that
adding
idea.
of a phosphate.
Sometimes it's taking it off.
Mm-hmm.
Yeah, it can take it either. It can be dephosphorylating it or phosphorylating. Yeah.
For the receptor, is that actually called receptor, or would it be called binding site?
No,
or
it's a
whatever you
receptor. It's a receptor.
Okay.
Usually a receptor protein. They do have a binding site, like the little site in the protein, but we just call
I
it
just wanted to
receptor.
know
Yep.
whenever writing.
Receptor. You could even call it the protein if you wanted to. You don't have to be. I am so ready for this to be over too. And then of course there's your little transcription factors. Remember, those are the things that kind of get transcription started. So that's just kind of a cute little cell signaling pathway. All right, to express or not to express. Here we are. So here, let's look at the box on the left. Those are your operons, okay? It wants to know, is this an inducible operon or a repressible? Oh, by the way, the top is like step 1 and the bottom is like step 2.
Inducible.
How come it's inducible?
Because the tryptophan guy is coming in, it's taking off the repressor
Okay.
instead of putting on a repressor, which would make it repressible.
Okay. Yeah, fine. In the absence of tryptophan, the repressor. Oh. Okay, yeah, you're fine. I think you're reading it a little wonky, but it's hard to read from there, so it's fine.
Is it tryptophan?
It's tryptophan, and tryptophan is also an amino acid, TRP.
Funny story, that's my uncle's name. No, I'm kidding, it's
Funny thing was, is I totally believed you.
I did too. I was like, oh, tryptophan. What do you call him?
What do you call it? Uncle Trip. Okay, so basically, I'm thinking.
Are you still thinking about?
No. I'm thinking about this operon because I just saw
It's kind of a-
I just saw an issue and I'm trying to figure out why that is. I'm trying to figure out if talking to you about it's going to confuse you more.
Wait, inducible's all like 24 off, like 7 off, right? Or off 24/7. So that's
Yeah,
why I'm kinda
actually,
confused.
this is not a great example because it's not a great example, but that's okay. According to the picture of the diagram and how it's worded, this is an inducible operon. Just don't write down on your paper that trip operon is an inducible operon because it's wrong. Because it's usually on all the time. I don't know why they have it drawn off in the first one. That's why I'm sitting there going, I don't know if I should. But this, according to this diagram, this picture, this series of events, this is an inducible operon.
So what would it look like if it was repressible?
It would be the bottom step would be first. Like that bottom one would be on top, and then the trip would come along and turn it off.
Oh, because it's off all the time. It's off and then it's turning on. So inducible's off to on.
Inducible's like able to be induced. So yes, it's off all the time. Inducible's off all the time, unless there's something that turns it on.
Okay, and then the TRP thing is turning on.
Yes, and a repressible one is on all the time, unless something comes along to turn it off. Yes,
Gotcha.
Adeline.
So in a repressible situation, a repressor is fine.
Yes, so if I wanted this to be a repressible situation, I would just take this bottom diagram right here, and I would put it as step 1. And I would say, here's our normal situation. There's no repressor. It's just the RNA polymerase is just making the proteins, or making the mRNA, which are making the proteins, and life is good. Like, we're just making it all the time. This is step 1. But then somebody comes along, locks into my repressor, which is like, locks on. By the way, the repressors aren't that far away. So locks onto the repressor, which would be around here somewhere, which then hits the operator and turns it off. That would be a repressible one because it's typically on. So all you have to do to switch this from being inducible to repressible is just put this one as step 1 instead of that one. Okay, so it's just like, what's happening first? Like, what's happening in a normal situation? Is it going normally? Is it always making the mRNA? And then, oh gosh, something comes along and turns it off? If so, that was repressible. It was able to be repressed, right? You can't repress something that's already repressed. I mean, it's already repressed. I can't repress it any more than it is, right? So it's not repressible. But if it's always off, our lac operon is typically always off, like not ours. The prokaryotes. Prokaryotes' lac operon is usually off. It's usually not making the mRNA to make the enzyme to break up lactose. It doesn't need to because it doesn't have it. And then if lactose comes along and there's no glucose, so we got to use it, then the repressor comes off and it's induced. It's turned on. Okay? Yep.
What's an operon again?
What?
Operon, an operon?
It's like a series of, it's like one of these. It's a series of genes. This is a piece of DNA, by the way, in case you're not sure what you're looking at. Piece of DNA that has a series of genes attached to it. They're in prokaryotes usually, and they'll make all those genes at once. See, they have gene E, D, C, B, and A.
Is that part of a transcription factor?
Transcription factor would be a protein that would The transcription factors are on the other. They're on the right side.
Are C and D being methylated?
No, they'll be expressed.
Are A and B also going to be expressed?
All of them will be expressed at once. That's why operons are super efficient. So the RNA polymerase will move across there. Bloop, and when it does, it makes all the mRNA for all of them at once.
So then what's
There's-
the difference between C and B?
What are you talking about? Are you talking about over on the right?
Yeah.
What the heck? I thought you were talking about genes C and D on the left.
Oh.
I was still talking about the operon.
Oh.
On the right-hand side is not an operon. The right-hand side is a gene switch. Okay,
Oh.
so this is if you want to watch something fun that teaches you about gene switches, but we already watched, but you can watch it again because what the heck is that Stickleback video. Talk about those little fish that grow those little spines, and it talks about how their spine genes are switched on and off, and that's what this is over on the right-hand side. The blue and purples and all those fun little bubblegum ball-looking things. That's a switch.
Isn't it like on because of environmental things? Like
It's-
it's turned on and off.
It can be. There's a lot of different things that can be. Environmental things can turn these on and off, or like hormones, or like signals from other cells. We have a lot of developmental switches. Like when you were being formed in utero, there was like switches. There were switches that were turned on during that time that said your head needs to go here and your butt needs to go here and your toes need to go there and all that stuff. And then they were switched off permanently after you were formed. And so we have those types of switches. You know, those are turned on by basically hormones. Lots of different things can turn on switches and turn them off. Depends on what type of switch it is. And actually our Stickleback ones, those are developmental switches too that they talk about because they're just forming the fish. Once the fish is formed, they can turn off. But basically what you're seeing here is A, B, C, D is time passing. Time A, time B, time C, time D. And by the way, that's super typical for operons, switches, anything to have a series of like snapshots. Like that's the first time and that's the second time and that's the third time. Like it's happening. These are a series of events. And so at first you don't have anything on your enhancer. It's just off basically. Do you see the inhibition line on top of the gene? Do you see that?
So then A, B, and C are being inhibited.
They're just not turned on yet. Yeah. They don't have anything turned on.
But C and D are being crossed? Or
Just
being
D, right?
methylated?
Just D. No, not methylated. Methylated is when the DNA wraps up around histones and shuts it off.
Okay, so then what's C and D?
Well, okay, so first of all, in A, it's just chilling. Nothing's happening. The DNA is just chilling there. Nothing's turning it on. And then in B, the process is beginning. You have two proteins that have bound onto your enhancer. Now, they could have been signaled from somewhere else or a hormone or environmental condition. We don't know. It doesn't say. Then your DNA starts to bend, and they're doing that because of the enhancer. The enhancer actually helps it to bend. Doesn't really matter. When the proteins that are on the enhancer come into contact with proteins that are on the promoter, or they kind of build a bridge, actually. When those actually physically touch, then your little transcription factors take off, and your gene is going to be transcribed.
Oh, so it's steps.
Not until then. Yes, in steps. So not until you've got them all bound together does transcription happen. So that's a switch. You don't really need to know the details of it. You should know kind of tell what's going on.
So enhancer binds to the promoter region.
Through a series of proteins. They have to have all those intermediate proteins to help them out because they can't really reach.
And the promoter region will let the gene transcription.
The promoter region's like the top box.
How long did it take you to like just answer all these questions without like any hesitation?
Good Lord. Years. This is my 7th year to teach this.
So first year you can do it.
I had to work really hard my first year. I was like, oh, it was so hard. I had to look up everything.
1 second.
Yeah, it's for real. There was a lot of, that's a good question. I'll look that up and get back with you tomorrow. There was a lot of that. Eukaryote on the right, prokaryote on the left is the rule, okay? Typically, prokaryotes don't have switches. Eukaryotes don't have operons. So we kind of see those that way. And I was kind of irritated because one year they made us count, not award the point if you wrote operon, because it was a eukaryote, when actually, let's be real, there are a few operons in eukaryotes, but they were not accepting it. So I would say, just know operons are for our bacterial friends, and we get the switches. Switches are way more complex. In fact, they think that some of those regions, like that enhancer region and all that stuff, are some of the introns that get chopped out. So
The enhancer region?
stuff like that, you know, because sometimes that'll be coded along with another gene. Like that's another gene up there somewhere. And it'll be encoded in the middle of it, and I'll chop that out. Okay, let's keep track of them. There you go. You've been wanting to talk about methylation this whole time. Okay. The green is DNA methylation. So DNA is wrapped tightly. So once again, we got step 1, step 2. So we're seeing a series of steps, right? Like this is a very common way for them to show time passing. So step 1 is up at the top. You notice there's no little methyl groups on it. See, there's no little black dots on it. Do you see that? But there's a binding place for them. And then down next step now the methyl groups are there. And then boom, the methyl groups and something else all wadded up and made that. And now we cannot, let's just say, is transcription off or on the first step? It's on. Get out of here. It's on. It's on, right? Because there's regions that can be coded.
Inhibited.
And then it's off at the bottom, right? Okay?
And in the middle, it's like on, but sort of.
It's on its way. Yeah, it's on its way to be turned off. And then on the other side is histone acetylation. See the little pink acetyl groups that are being added to it that are rolling the histones away from each other, which turns your transcription on. Nice, that was anticlimactic. Is transcription off or on that one? Turns it on, and it fades into nothingness. It never tells you the answer. Maybe the answer will show up. Nope, didn't. Okay, so that's your 2 kind of ways to turn genes on and off. Any questions about that? Anything you want to talk about on that? Epigenetics. All right, then we have our, what happens? Oh, never mind. JK. Oh, no, just kidding. I'm not giving away anything. Stop talking. What is pictured here? Transcription. No, not ribosome. This is transcription, right? How did you know? Somebody who knew. Tell me, what was your giveaway? Help each other figure out what's going on. Okay, Shannon, what?
The template strand is coming in and it's being coded.
And what's being coded? Like, how'd you know something was being coded?
With the RNA polymerase.
Okay, you had RNA polymerase. That was one clue. What if it didn't say that? How would you know besides that?
Because the DNA is in
You got the blue DNA is opening, and what's coming out of it? Yeah, you got that red thing. There's your kind of giveaway. You're not going to see that in DNA replication. You don't have anything like leaving it, right? Okay, so that's transcription. Where does this take place in prokaryotes? Nucleoid region. Where does this take place in eukaryotes? The nucleus, okay. What's that? No, the red
No, it's the mRNA.
mRNA. Why is it called the coding strand up there?
Because it's going to be the same as the other strand because it's the one that binds into template.
Good. Adeline's on fire on this one. She's like, I might have got confused on another thing, but this I got solid. The coding strand, because it's the same code. It's the same code as the mRNA will be, minus the T's and U's. But otherwise, it's the same code. So, all right. Can you determine if this is prokaryotic or eukaryotic from this picture?
It's
I don't know. How can you tell?
because translation is happening, it happens simultaneously.
Oh, I see. So you're saying if this was prokaryotic, we should see like a ribosome down here. Okay. All right, cool. I'll take that. Otherwise, it's hard to tell. What's going on here?
Introns are being spliced out.
Why are
Prokaryote-
the
what?
introns spliced out again?
Because there are non-coding regions of DNA. So if you were to send them to the ribosome, they would not, your protein would be wonky. It would have big chunks of weird stuff in the middle.
Eukaryote.
Okay.
Because prokaryotes don't have introns.
Pro-
That's weird.
Prokaryotes don't have introns. Like I said, Dontor and I were talking earlier, and I said they have to pack light. They only have one chromosome. They can't be adding in a bunch of weirdness to it. Okay. Let's take a look at this for a minute. So this is a little diagram of alternative splicing. So you see these blue, green, pink, teal, and orange at the top. Those are your exons. Those are your coding regions. And in between it, then you have your introns. And you can see that I can make 3 different proteins out of this same sequence just by splicing out some of my exons and doing it differently for each one.
What
So
are the arrows again?
those arrows, is that what you said?
Yeah.
That arrows are just like saying it's example of alternative splicing, and then translation means then it goes to that.
Wait, did you mean introns?
Did I say splicing out the introns?
You said
Well,
extrons.
well, actually, it's spliced out exons here. Like right here, see? Blue, green, pink, teal, and orange. So all 5 of them are in this one, but this one is spliced out number 3 for some reason, because it's an alternative splicing.
Where are the arrows like on the protein?
Oh, those? Oh, that's just showing you a direction of the protein going. I have no idea why they put those on there. Something that biochemistry people could tell you, but I don't know why they do that. They do that quite a bit though. It's almost like a directional way the protein is folding, but it doesn't seem to matter to us. Is that what arrows you're talking about now?
Yeah.
Oh, I thought you were talking about these arrows.
Oh, okay.
This is just like alternative splicing happened and we got this, and then translation happened and we got that. But you can see they're splicing out an exon. So they're actually splicing out a coding region. 1, 2, 3. This one's missing number 4. And so then you get something a little different. Are they similar? Sure, they're similar. They're just sort of missing a piece here and there. And why do they do that? It's another form of gene regulation. We can make a slightly different protein just by doing a funky little splice instead of creating a whole nother protein. That's amazing to me, actually. Kind of blows my mind. What step does this represent?
This is
Translation.
ribosome, right?
Dang it, me in Brooklyn. All right, so translation. I hope my straw hit the floor. There's no telling what's over here. So translation is basically now we're turning this thing into a protein. So hey, Ally, maybe this will help you, because this is a diagram of what we were talking about a minute ago. So maybe this will help you kind of envision what's going on here. What's the yellow one? What is that thing? Mm-hmm.
Nuts.
It's an amino acid.
Is it really?
Uh-huh.
Oh,
Amino
It's his
acid.
uncle.
It's your uncle. Duh. Why don't you know that? What is this one here, this blue one? That is your tRNA. So, and I accidentally already, oh man, I took that one off and I didn't note it. The orange one. Did you guys see the orange one?
No, I didn't.
That's
But I'm
the
gonna guess
mRNA.
codon.
Well, look at what it's pointing to. Follow the line. It's a codon.
Oh. Yeah.
It's pointing to a codon. Okay. So a codon is the purple and it says AUG, which codes for methionine. Do you see the little tRNA sitting on top of the codon area there? You see it? So it brought in, so the tRNA here is bringing in this amino acid. This tRNA brings in this amino acid. And basically it's like, hey, this is mine. A pairs with U pairs with A, C pairs with G. And so I'm going to leave methionine right here, which is good because it's the start codon. And so then that methionine will get stuck there and then the tRNA will go away. And the next tRNA comes in and he's like, oh, hey, this is mine. A pairs with U, C pairs with blah, blah, blah. And then this TRP will be attached to that MET and make like two little balls together. And then they'll keep going and going and going until it makes its whole protein. Okay? So that's how that works. So the anticodon is on the tRNA and the codon is in the mRNA.
Where does the top box play a part in this?
It doesn't. The top box is in the DNA, which is where that mRNA came from.
Wait, what does the Tata box do again?
Top box is a promoter region. Promoter region of the DNA. The promoter region is what tells it to start transcribing. So the Tata box plate, let's back it up. Back it up. Oh my gosh. Okay. So see how it's transcribing here? This had to know where to start.
Mm-hmm.
And so there was a promoter region somewhere on this DNA, the blue stuff that's open.
Can you show me exactly where it started?
Can't show you on there, but I think I can, maybe right there. We can pretend like it's an operon. I don't know. There's a promoter on the one over there. So like if this is DNA right here, then we want to transcribe this gene, right? Because then we're going to make the proton. They're going to make the protein out of it. And so we want to transcribe this one, but it's off, right? Well, here's a promoter region. Once my transcription factors get on the promoter region, it doesn't have to be a switch. It could just be a normal DNA. This is special, right? This is a gene expression, a way it can be done. It can also be done just with a transcription factor sits on a promoter region. This would be your Tata box right here. your promoter region would have a transcription factor protein that would sit on it and say, hey, I need the RNA polymerase. There it is right there. Okay. Once that's there and everything is all connected and the signal says go, then it can begin to transcribe the DNA. So that's what that is.
So what was the thing on the top box?
Transcription factor.
Okay, and that is a range
Oops.
of things.
And so transcription factors are proteins. And so then basically back here somewhere, so back up the polymerase, like rewind the polymerase. Back here somewhere when it was unwound, back then there was a promoter region somewhere. The transcription factor sat on it, recruited this huge RNA polymerase, which then took off and began to transcribe your mRNA.
And that transcription factor is the promoter?
It sits on, no, it binds to the promoter region. The promoter is actually a region of the DNA, TATA, like thymine, adenine. But then, of course, there'll be a whole bunch of other things. So it's like TATA, AAATT. They're different for each gene, basically. They have a whole combination of A's and T's. And so once the the transcription factor sits on the promoter region, then the RNA polymerase is recruited to begin. So then here it goes. It makes your mRNA, splices your mRNA, and then now your mRNA is down here, nicely spliced and ready to go. Okay, yeah.
How did the mRNA go from like rRNA to tRNA? Like, is that just a range of steps?
mRNA comes from your nucleus. It is formed from the DNA. rRNA is the ribosome. So mRNA goes to the ribosome, usually by a transport protein that carries it there.
Okay.
So mRNA leaves the nucleus, goes to the ribosome, which is the rRNA, ribosomal RNA. And then tRNA is right here and comes in and meets up with the mRNA. Okay, so they don't go together.
tRNA is being in the ribosome regardless?
No, it's just floating around. They hang out over here. tRNAs are outside of the ribosome. They only come to the ribosome whenever there's an mRNA to be coded. And that's all a process of signaling. They're just floating. They're just floating around, hanging out with their amino acid buddies, waiting for the party to start. Okay. Any other questions about that process? That is a super important process, by the way.
That part is so confusing.
And it's unfortunate it is the central dogma of biology. So you kind of need to get at least need to get a small grip on it. Yes.
So like, the tRNA and RNA are just like, they're not coded for.
Well, they're coded for in the nucleus. There's another region for them to be coded, but we don't ever talk about that in our class. We just assume they like magically fell out of the sky. But DNA actually has codes for rRNA and tRNA as well. I can't tell you where they are on the DNA. They're somewhere.
And then they just like travel to the
And actually, you can be born with some of your RNA and stuff because you can get some of it from your mom because they do you do get the cell from your mom, but for the most part, we just code out new rRNAs.
It'd be so easy to learn if we could just like magic school us, in there.
Can you do that?
No, I haven't got that ability yet. I'm working on it.
When you figure that out, let me know.
We need to upgrade. I need to upgrade your Teacho. You have to go bye-bye? You have to go bye-bye?
I have to go to track practice.
How much longer?
She's got to go practice for state, for real.
How much longer do you have this?
Oh, I don't know. This thing? I've got hours of review I could do. No, I have no idea. We'll find out. Good luck.
Thank you.
All right.
Yeah, we have basketball track.
Oh, I know you do. So you'll just go when you need to go. Whatever. So there's your codon chart that you don't have to memorize. Thank goodness. Can you imagine? I'd probably drop out. And so then we're going to talk about mutations. Okay, so if you have a point mutation, what is that?
That's just the same one. That's mutated.
All right, so silent mutation has no effect on the protein sequence. See that top one? So do you see the very top one where it goes, it's coding for serine, valine, proline, and is that tyrosine, I think? Anyway, and so see how even though there was a mutation in the DNA on that sixth nucleotide, that second codon, and didn't change it at all, right?
It's the same.
Still, it's the exact same thing. Codes for the same thing. So that's a silent mutation. What is one that results in an amino acid substitution?
Point.
It's a point, but we call it a missense. Doesn't really make sense, but it's okay. Why doesn't it make sense? Why would that be like what am I talking about? It makes sense. Like, what am I talking about?
Like missense and substitution.
What are we making here?
A different protein.
The different protein. And it may not be a completely different protein, but it may just fold weird, right? And just kind of doesn't make as much sense. Like, maybe it doesn't work. Maybe it doesn't work great, but it still sort of works, so you kind of see some functionality, but not exactly the kind that you did, that you expected. And then what's the one that goes for a stop codon? Do you remember that one? Nonsense. That literally makes no sense whatsoever. It's like you were going along and you were making this really nice long protein, and all of a sudden, boom, stop. You only get half your protein, and that thing don't work at all. Doesn't make any sense at all. What about this bottom one, this letter D? It says insertions or deletions of nucleotides may result in a shift in the entire reading frame, or insertion of a stop codon. It's somewhere weird.
Frame shift.
There's your frame shift, and those suckers are a hot mess.
That's when you don't get a good
You can get all kinds of weird proteins out of a frame shift. Okay. So I think we have a pretty good handle on how all that works, right? And all this is a product of how it's going to fold, and I'm fixing to put the protein folding up there. All right, so polypeptide bonds. They're peptide bonds. Dehydration synthesis forms these. We've talked about that already. So there's your little, oh gosh. Oh, this is the one that has the R groups on the top. You see them?
Mm-hmm.
Flipped it upside down. And so they've bonded 3 together, and so it's in the process of making a little protein. So that's your primary structure, right? Everyone cool with that? And then secondary structure is you're making little hydrogen bonds between your groups. If you get a mutation in this, it can change it slightly. Your secondary structure can be changed slightly, but it probably won't see that big of changes. This is when you get things like alpha helix. Oh, alpha. I mean, yes, it can be alpha. Alpha helix, which is basically just a kind of a twirling of your protein.
Yeah, it's in the secondary.
Secondary.
Twirl.
See it? So a little twirl looks kind of like a DNA, but that's not a DNA. That's a protein. And then the big one, the one that we really talk about the most, is your tertiary structure, which is this green one here. And that's your R groups. The properties of the R groups drive these. You get interactions between the R groups.
And that can change the amino acid, the R group.
A change in amino acids can really change the R. Yes. Okay. So
other way.
So the R group is in your amino acid, right? And so if you swap it out with a different one, then it can really change your protein structure, right? Mm-hmm. And so here's a little picture, a little drawing sort of how it kind of works with different we say interactions because they're not all bonds. There can like it could just be hydrophobic, hydrophilic. That's not a bond. That's just an interaction with the environment. It can be an attraction between positive and negative. That's not technically a bond. It's just an attraction. And that makes the shape of your protein, which makes the function of it, which is why it's so important. Yes.
So if a question was posed, there's a point mutation on the polypeptide chain.
Mm-hmm.
What structure would it affect? Would we say that it would affect secondary and tertiary?
I would. Tertiary would be my favorite one. I'd want tertiary in there for sure.
Choice,
and there was one. I know what you're talking about. Yeah. So it typically won't affect your primary structure because your primary structure is literally just a chain. Now, if you have a frame shift, that can affect your primary because it can make it short, right? So now all of a sudden, instead of 250 amino acids in a row, you only have 210. Well, that just affected your primary structure because your little train doesn't look the same anymore. But if you're just flipping one out, it's like flipping out a train car. It's like, did that really affect your primary structure? I mean, not really. You still have your train car. You still have the same number. The secondary structure, you can see some difference in your bonding depending on your amino, but you're not going to see a whole lot of difference in your secondary structure, but there's potential for some difference there. So if I wanted to be specific, and if the question said, definitely tertiary structure. Yeah. So absolutely tertiary. But secondary can be affected too. And of course, quaternary can be as well if they put it on there.
Wait, why tertiary though?
Tertiary because if you have a mutation that changes an amino acid and it switches out that R group, you could have a hydrophilic or you used to have a hydrophobic. And now your
Oh.
protein is like jacked up and your active site screwed up and like you're like, what the heck just happened?
So the last one's folding.
The last one is the quaternary, which is just like see I have an orange and a brownish orange and a pink and a sort of purpley pink. Those are the 4 different like little, I don't know, subunits of the protein. And when they are like kind of attracted to each other, it makes up one big globular protein. Not all proteins have a quaternary structure. In fact, most of them don't. It would just be one that had a certain specific activity that required that. Hemoglobin. Hemoglobin has quaternary structure. Okay. Ta-da! Where are proteins going that are made in the endoplasmic reticulum? I said there are 4 different places you could say. I expect you should at least know 2, maybe 3. Okay, so where are they going if they're made in the ER?
Getting out.
They're going outside. There's one. Huh?
Sorry.
The cell membrane. I hear people whispering. I'm not sure why. So they're going to go to the Golgi probably. A lot of them are going to the Golgi, and then on from there, you could go to the cell membrane. You could go outside the cell. You might be just staying in the endoplasmic reticulum. I don't know. Maybe they need you there. Also, lysosome. Oops. Ta-da.
Oh, wow.
Because lysosome's the one that has that acids, that they're super acidic, so you can't just drag that through the cell. It would just ruin everything it touched. And then what if they're made in the free ribosomes? The ones that are just floating in the cytoplasm. They're just used right there within the cell somewhere, okay? So they're just going to be used in the cell. They don't pass with the Golgi. They just go wherever they go on.
Wait, so then where are those ribosomes created?
Created?
Yeah.
There are RNA, so the ribosomes are created in the nucleus. I mean, they're made out of RNA or RNA.
Oh, so the ribosomes are created inside the nucleus?
Yeah, but we don't need to worry about that. We're worried about where the proteins are going that are created in the ribosome. The free ribosomes that are just floating around in the cytoplasm, the proteins that are translated there are just going to the cell somewhere, not the lysosome.
The Golgi just really makes things go where they need to go.
I love the Golgi.
Right?
Yes,
It's
its whole entire job is
got-
to make sure everything goes where it's supposed to go.
I thought that was the mother goose of the cell. I like it.
The nucleus only. So we learned, back in 7th grade, the nucleus is like the command center, the control center. Okay. The reason they say that is because it has the DNA, and the DNA creates proteins, and proteins literally do everything in the cell. So, but the nucleus isn't like going, this needs to go there, that needs to go there.
It's like the
The Golgi does that, yeah. It's like the UPS or the Amazon facility, I guess. We used to say UPS, but you guys don't know what UPS is. What am I talking about? Oh, okay. So we don't need to talk about this too much. It's the promoter region there. We talked about this yesterday. So anybody need to go over this again? We went over that yesterday.
that was the most complicated lab.
It was so complicated.
that lab.
It was good.
I
So
had
much
to
of
like really.
I had to watch videos about
You're
people
like
doing it, so I
on
could
the-
understand what was going on.
I still know.
That lab was so complicated.
Well, it's
Yeah.
because we didn't have like
Uh-huh.
time in class.
I know. That's because we were on stupid hybrid schedules, so I couldn't sit there and talk about it. I'm like, watch this video, do this, fill this out. And I'm like, oh my
Wait,
gosh.
what does the minus mean?
It means that they don't transform. They don't have the plasmid. The minus sign means no plasmid.
Was this the lab that we did for ecology?
What, Steven?
Was this the lab that we just did for ecology?
No, this was the lab that you did with pretend like virtual micropipettes that you had to put stuff into little microtubules. You did a bacterial transformation. You can find it in in your Canvas modules. It would have been Unit 3, I think, for the semester. But you did one of these. You guys going to basketball tryouts?
Yes.
Good luck. May you both get your whatever positions you're looking for.
Bailey wants to post.
Of course she does.
I'm going for post.
She wants to be
Rowan's gonna be
she
the
wants to
point
be
guard.
that tall person in the middle. Okay. Do you like how I know my basketball positions? She
Yes.
wants to be that tall person in the middle.
Yeah,
It's okay.
That's
But now, like, they're all tall. There's
Oh.
so many players who are like 6 foot now. Well,
Yeah, you just gotta be
Bailey's working on it.
faster than them, Bailey.
yeah.
Faster and
Bailey's
stronger.
got the skills.
All right. Yeah, Stephen, this was in unit. We did this lab. We should have virtually. It's basically making bacteria glow.
Okay.
Wait, is this the one that Ryder's completely messed up?
There was one group in 7th hour that completely messed it up. Yeah, good job. You go down in history as the one group that messed it up. Otherwise, we had 100% transformation this year. I don't care about this. It was just a list of proteins, things that can be receptors, enzymes, hormones, structural. It's just a list of things proteins can be. Okay. Yay. Enzymes, vocab word. And you guys, when you need to leave, just leave. I can just stay here and talk to myself. It's all good. I'll just stop when the last person leaves. Vocab word. Give me that first one.
Active site.
Hopefully that's what pops up. There we go. Active site. I'm like, I really don't know what order these are going to come off. And then what's the yellow thing?
Substrate.
Substrate. Some ugly pictures. They're reusable. They can synthesize or hydrolyze. Ooh. Hydrolyze. What does hydrolyze mean? Lyse. What does lyse mean? Take apart. I don't know what the hydro's doing in there, but anyway. They can hydrolyze. They can break things apart. They can synthesize things. They have an optimal pH and temperature. Like what their optimal is whatever they're adapted to. Cold doesn't denature. It only slows it down. Heat can speed it up, but get it too hot and what happens? What does denature mean?
It doesn't work.
Like falls apart.
Right, all those little bonds that are holding that active site into its shape just get loosey-goosey. And then the active site isn't the right shape anymore, and then bazing, doesn't work. Oh, did you guys need that? And then heat and pH, either high or low denature. Either high or low pH. Denatures at extremes. Okay. This is an activation energy graph. The blue is with an enzyme because you use less energy, right? The red is without an enzyme. It still might happen. It's just going to require a lot of energy. But notice the products. Tell me about the energy of the products. It's the same. It's less than it started with. So this is an exothermic reaction, right? Because it released energy. But they're the same with or without the enzyme, right? So it's not like it changes really the products, really. It just changes how fast they get there or how much energy has to be used to get them there. So that's what enzymes are for. We have enzymes all over the place. We have enzymes that make our DNA replicate, right? Like all those helicase, all those polymerases, those are all enzymes. They have an active site. Stuff fits in it a certain way. If they're there, it works. If they're not, basically, I guess we can make it happen, right? We can make DNA replication happen without enzymes because we can stick it in a PCR machine and heat the heck out of it and make it fall apart. But that's a lot of energy. We don't need to use all that much energy. Just throw an enzyme, Our cells don't have to use that much energy. Can you imagine? It gets up to like boiling. You have to get your cells to boiling to replicate your DNA. I feel like that'd be bad for everybody involved. I don't want my cells to be boiling. Something blocking the active site so the substrate cannot bind is called what kind of inhibition?
Allosteric.
Competitive. Competitive. Down doors. Competitive.
Can you go back there? Can I write that down?
Something blocks the active site so the substrate cannot bind is competitive. Because it's competing for its active site, right?
But then, would the active site be blocking?
Do you still need it, Anya? Sorry.
Cannot. Yeah, good. Right, good.
Would the active site be blocking it with allosteric?
Allosteric changes the shape of the active site. So there's not something competing with it. There's something that's turned it off. I like that little cartoon. It's probably in my bag over there. I have a whole bag covered in biology cartoons. Something binding to a different site, which changes the shape of the enzyme. There's your allosteric or your non-competitive, whatever you want to call it. Allosteric. I'm just like, I'm going to take a picture so I can write this down at my own time. Yeah.
Well, let's look at this. There you go.
Well, just remember the competitive one's actually competing for the active site. It's like two things, trying to get the active site.
Why is it also called allosteric and not just non-competitive?
Well, I think, I'm not sure what the root words are or anything like that, except for allo means different. And so it could be like it's a different site. But we still call the place the allosteric inhibitor binds the allosteric site. And so that's why you kind of want to know it both ways. Because it is non-competitive inhibition. It's inhibiting it without competing. But the site where it binds is called the allosteric site. And basically that just means remember we did geographic, whenever we did what was it? Oh, it was like sympatric and allopatric speciation, where sympatric's same country and allopatric's a different country. This allosteric inhibition, it's a different place. So that root word is different. So it just means it's inhibiting in a different place.
Allopatric's only one is geographical, right?
Yeah. So allopatric. And they're showing you here something I never really talked about, but it's in your textbook. They're showing you allosteric activation, where instead of inhibiting it, it's actually making the binding site available. So that could be a thing too. Like you could have something come along and walk into an allosteric site, and instead of inhibiting the reaction, it's actually causing the reaction. So that's a possibility. I don't know. Just kind of, these are critical thinking things. This is when you know the basics of it. You just have to go with what they're giving you and work with the basics you know and build on it. Because there's no telling what they give you. Yep.
So we were saying allosteric is outside. So we're having another thing going in there that's changing the shape of the active site, right? Then what would be, how can you have something not come out and change? Like you're saying non-competitive inhibition
It's the same thing as allosteric inhibition.
is the exact same.
Yeah, it's just a different word. So the allosteric site is the place where the thing will lock into, and so it's called allosteric inhibition or non-competitive because it's not competing for the active site, it's just changing the shape of the enzyme. Okay, so feedback. What kind of feedback is this?
Oh, negative.
It's negative. Why?
Because there is a stop and an end and a cycle.
What? Yeah. What
It's
is stopping?
homeostasis.
What is stopping the process?
The end product.
The end product. The end product of the process is stopping its own process. That is negative feedback. Because if something is being built, so the little yellow box, see how, so substrate can go either way. Green substrate locks into the little Pac-Man looking enzyme, right? And then enzyme does that, it goes to that. It's like a little pathway here. And then boom, enzyme 3 makes a product from that. So basically probably breaking up the blue thing into a yellow square and something else. So there's your end product. And then when the end product comes over here, it inhibits the whole pathway. That's negative feedback.
So if the end product, like if the yellow square wasn't there, would that automatically make it positive?
If the yellow square locked onto the enzyme and made it go again, that's positive
So if
feedback.
like that red arrow that's right now at like an angle.
It's bouncing out.
If that wasn't bouncing
Yeah.
out, it would be positive.
Yeah, if this yellow box came around here and locked into that one, it would be positive because now it's making it go over and over and over again. But since it's stopping it, it's negative. So if the end product stops it, negative. If the end product makes it go, positive. Sometimes the end product doesn't have anything to do with it at all, and then there's not feedback. And it's just cell signaling. Okay. Cool. Why is the rate of the reaction of the competitive inhibitor higher than the non-competitive?
Why is the rate?
Because in the competitive inhibitor, there's like an equal chance of like the substrate and the inhibitor going to the active site, but with the non-competitive inhibitor, the substrate doesn't have a chance of competing, and as a result, the
Yep.
non-competitive inhibitor
Well-
has a lower rate.
Well worded, yeah. As long as it's a competition, you've got a chance to win, right? So if it's a competition, the substrate could make it there and get whatever happens first. But if it is non-competitive, it's changing the shape of the active site, you don't have a chance. Like there's no chance. Once they bind in there and stop those and change those active site things, you just aren't going to be able to see it. You're not going to be able to have any product at all. So, but maybe before they get it inhibited, a few of them can be made. But after it's inhibited, no chance. Hey, what's an independent variable here? Whoa, bounce back to some experimental design.
Substrate concentration.
Okay, substrate concentration. Yes, there's one. The inhibitors are also ones, right? Your non-competitive inhibitor and your competitive inhibitor. I don't know what the red line is. Maybe the red line's like normal. I don't know. Without any inhibitors. I don't know. Did it tell you?
Why do you have a non-competitive inhibitor?
Why have one? Cells will create them if they need to turn off a reaction and turn it off good. If they need to shut down a reaction, a cell will produce a non-competitive inhibitor. Or
So it'll just bind in there. It will inhibit completely, not and then competitive. It will be
Yeah. Yeah, and also this question of why is hard to answer in biology because it could just be that's what evolved, and so that's what we got. But also you can see these in medicine. Like people will chemically in the lab produce non-competitive inhibitors to shut off reactions so that it'll change something medically for you. That's kind of a thing that happens too. What would be a negative control?
No inhibitor.
No inhibitor, yeah. No substrate, maybe. No, no whatever it is. No enzyme. Yeah, don't put an enzyme in there and see what happens. So take out one of your research subjects, right? And actually, a no inhibitor might feel more comfortable just being a control, like a regular control. I've got a competitive inhibitor, I've got a non-competitive inhibitor, and then my control. I'm going to compare that to not having an inhibitor at all, and we'll see how they do. I mean, you could do that. Definitely, if you left out your enzyme, you would have a negative control, right? You expect nothing if you don't have an enzyme. Now, if you get some results, you got something screwed up in your experiment. Yep.
So is there such thing as a positive control?
Positive control in this one would be something that we know would have an effect. I don't know. If I was going to do a positive control in this one, I'd have to know more about what we were doing. There'd be something that we could add to it that I know would work. And if it didn't work, then I'd be like, oh crap, experiment's not working. Control group, we talked about that. Okay, and then meiosis for different cells. Just kind of be able to recognize it. Make sure that if they ask you what's the end product of one cell cycle of meiosis, to say 4 genetically different cells. Make sure you contrast it with mitosis if they ask you. I don't see that very often. It was on our mock, but it's not a super commonly asked question. What the end product of mitosis or meiosis are.
And it's the 2 identical.
Mitosis would be 2 genetically identical. Meiosis would be 4 genetically different. So, like, you need to differentiate between the 2 processes.
Meiosis is 4 genetically different.
Okay. And they're half cells. They're gametes. Okay, and then what's going on?
Are all gametes haploid cells?
All gametes are haploid cells.
And all mitosis
It's diploid.
are diploid?
Yep, crossing over, recombination. Okay. And then of course we're just plowing right through, we get into genetics. So there's your 2 haploid cells on the left, put them together, they make a zygote, which is diploid. That's showing their 2 little nuclei getting together, and then the next scene, which it doesn't show, those nucleus have formed, and formed, good Lord, fused, and have formed one big nucleus that has all of the DNA in it. Okay, so that's how you get your fertilization. How come there's no allele on the Y? It's a boy. Why does the Y not have an allele though? That's not fair. Huh? It's sex-linked. It's carried on the X. The Y is not an X, right? So it doesn't have one. Oh, we didn't do those because they're not in your standards. Sex-linked trait, we got that. There they are. Multiple alleles, different alleles. Dihybrid crosses. This is all just genetics. What kind of inheritance is this? Autosomal recessive. I agree, Caitlin. Why did you say that?
Because they make kids that don't have it, kids that do, and then the 2 kids don't have it, they all the black make all blacks.
Yep. But what about these? This 2-1 and 2-2 here.
Yeah, if you look at you have to have a.
You're right.
Because they can still be carriers.
They can still be carriers, right? So 2-1 and 2-2 must be carriers because they had 3. No way could 1 and 2 be recessive and have a dominant kid. Like, no way. Okay. Pedigrees are like the ultimate, critical thinking exercise. Like when you get them, you just have to sit there and go, okay, what is this one? What is that? That's just all they are. Like I try to train my pre-AP kids or my freshman kids how to read them, but in the end, it's like you just have to sit there and figure it out. And once you figure it out, you got it. But it takes a little bit of time to kind of figure it out. I got a couple of them here. Anybody got a guess on this one? Sex-linked recessive. I'm going to say sex-linked recessive. We got a bunch of males that have it, right? And it's definitely recessive. I mean, because you're not seeing it very often. But if it was sex-linked dominant, how would I differentiate that?
Bunch of females that have it.
The females would have it just as often as males, right? So that would. What if it was carried on the Y? What would I see then? Only boys, and it would kind of look mitochondrial except for males, right? So it'd be like if that 3-4 guy has it, right? His son would have had. Well, he doesn't have a son. Oh yeah. So never mind. Bad one. Do we have any like
Wait,
that?
so
Yeah.
autosomal
So if
recessive?
4-8 had, all of his kids would have to have it too if it was Y length. And we never even talked about Y length, but you can figure it out. I mean, same thing as that. What?
So then if it's autosomal dominant, then there's gonna be an equal amount of males and females that have it?
Autosomal is.
Oh wait, no, sorry. Wait.
Autosomal is anybody's
Sex-linked
guess. Sex linked, you could have males and females. It would be the same because you only have to have one to have it. And so if your ex has it, then and you know if you have 2 exes, doesn't matter.
So then will most of them have it?
Yeah, but I don't think they'll ever put a sex-linked dominant on because it looks just like autosomal dominant. Like you can't tell the difference. Okay, what's going on this one then? I don't even know what I put on here. This one of those impossibles to tell because they're my favorite ones to put on.
Autosomal dominant.
It probably is autosomal dominant. I don't know. Kind of weirds me out that one person 2-1 has it. None of their kids do, but. Oh wait, yeah, that can't. Well, it could be. Could be. Yeah, screwy. I hate it.
Could it be
Okay.
mitochondrial because the mom?
Oh, is it? Is it mitochondrial? It is. It is. We forgot to look for mitochondrial. You are amazing. I should put a little "you're terrific" traffic count on your head. I have it somewhere. I don't know where it is.
It's right there.
Yeah, I should put that right on your head because you're amazing. That's mitochondrial. Good eye. Good eye. Okay. Tay-Sachs. He's got a screw up. Do you see it? Do you see the mutation there? So, and there's your little thing, and there's all the good stuff, and then it throws on the which one is Tay-Sachs pedigree. It's recessive, by the way. Tell me how you can know. Okay, what is Tay-Sachs? Do you know what Tay-Sachs is?
No.
It's that disorder that they die when they're like 3. So if they die when they're 3, can they have children?
No.
No, so it's got to be B. I put that on there because I was mad about that question that was on the test. Well, notice the ratios. What's happening here?
They're sex.
Let's see. Are they sex linked?
No, not linked. They're linked.
They're linked genes. They're linked genes. Which ones are linked? Are both of them linked? Yeah. No? One of them is linked. Which one? The one on the right or the left?
Right.
The one on the right is linked. What does linked mean again?
They're really close on the same.
On the same chromosome. Could you calculate the recombination rate there? Do you know how?
Wait, why is the one on the left not?
You'd add.
The one on the left looks like a 9:3 to 3:1 ratio to me.
Oh, okay.
Would you add 76, 79 and then divide by the total of
Yep.
all those?
You add your recombinants together, so you add 76 plus 79, and then you divide by your total number of offspring, however many that is. And I think I'm fixing to stick it on there again. Recombination rate, there you go. Should be 9% or 0.095. Crossing over, that's how you get your recombination. Karyotype, is it a boy or girl? It's a girl. And, oh, geez, switching gears. Anytime you guys are like sick of this, Miss Wilhoit, you can take off. Switching gears, now we're going into cells and transport. So, what is able to diffuse through the membrane freely? Yeah, Elena.
Non-polar small molecules.
Non-polar small molecules. So something that's not polar and small. Good. Not charged. Active or passive? Regard the diagram. That's active. How do you know it's active?
The lower arrow is making.
Well, you're right. The little arrow is probably our ATP, though it doesn't say it. I think I took it off so that it wouldn't give it away. How else can I tell? Yeah, it's having to stuff into a high concentration area. How about the one with green blocks? That could be passive. Yes, absolutely. Okay. There's your water. It's bonded together. Covalent bonds. What is that protein called if that is water?
Aquaporin.
Aquaporin. Water is polar, has to have a thing. It can't sneak through. Oh, now we're at the top. Hypertonic was the one that shrunk. What type of solution is the middle one in? ISO. And the top one? Ooh, what happened to it?
It got bigger.
It popped. What do you call that? What's the L word that I say shows up all the time? It lysed. Yes, it lysed. Good.
How you spell lice? Is it
L-Y-S-E. Yeah, lyse. And you see that all the time. You know, hydrolysis, that L-Y-S-I-S or whatever. It's lysis all over the place.
Lice is burst.
In that one, it just means it burst. Lysed. Yeah, well, lyse just means break, is what lyse means.
So lysosomes break down.
Yes, lysosomes are the organelle that breaks stuff down. Yes. How are the cells below different from the ones above? Why? And they're the same thing. They're in the same solutions. Like, what's the deal?
Cell wall.
Yeah, the cell wall keeps them from lysing. Okay, which end of that arrow on the right-hand side, which end of the arrow is more negative value for water potential? Which end is going to have the lowest number, the top or the bottom of the arrow?
Oh, are you talking about the tree?
I'm talking about, it's got an arrow beside it.
Okay.
Yeah, on the whole right-hand side, I'm just reading that green box. Which end of this arrow has more negative value for water potential? Which end is the top? Why?
It says low,
Tell me in biology why.
because there's less water.
Capillary action can only really get you so far.
So negative.
Yeah, so it's got to move towards the negative, right? Because like Steven just said, capillary action that moves through the plant, it can only get you so far. You have to be able to have some water potential involved so that water will move, because water moves towards the lowest water potential, right? So it's like a sink. Like the lower it goes, the more likely water is to run in that direction. So you're going to have some way to get that water all the way to the top of that tree. Like it's going to have to fight gravity the whole way up. So what they do is they adjust. Remember how I said you can put turgor pressure in that vacuole in the middle, and they can increase the pressure, decrease the pressure, so they can actually help their water potential along. It's kind of cool. The lowest would be your atmosphere.
Wait, what's water potential again?
Water potential is the potential of water to move into an area. We calculate that with like the little trident thing. Okay. And then here's, of course, what do we have to get into now? Holy cow. Can you believe you can like take a second, look at this and sort of know what's going on? Like that is insane. This is a weird looking one. So this is in your thylakoid membrane. That's the membrane, the little yellow dots and stuff are the membrane of the thylakoid. Yeah, what's going on here? We got PS2, we've got our electron transport chain, you've got PS1, got all kinds of good stuff going on here. So what is this process here?
Photosynthesis.
Photosynthesis, light-dependent reactions, right? Light-dependent reactions phase of photosynthesis. What is the purpose, the end goal of these light-dependent reactions? We're producing energy, ATP and ADPH for what? The Calvin cycle. Calvin cycle, right? Okay, where are you going to find chlorophyll in this particular diagram?
680 and P700.
And the PSs, yes. P680 and the P700, he's reading the diagram. PS2 and PS1. There's your chlorophyll. And the purpose of the electron transport chain?
To turn on the proton.
Got to get a proton gradient, right? Getting a proton pump turned on. You got a proton gradient building up in the middle of the thylakoid. And what would happen if the thylakoid membrane became leaky? The favorite one, so I put it in red. What would happen if there's little holes in that yellow thing and your protons are just leaking out?
ATP wouldn't be created.
Right, you wouldn't be able to make ATP because you wouldn't have protons to go through there to make your ATP because they'd all leaked out, okay? I don't know why that's a favorite of theirs, it just is. Alrighty then, what's your photosynthesis? There's your Calvin cycle. There's your, what stage of cell respiration is this representing? This is glycolysis. All right. So here's your cell respiration in a little train. What if that is broken right there? What is that? Acetyl-CoA. What if it's not working?
Then fermentation occurs.
And then I guess, yeah, you'd shut down the process and you'd have to fermentation. So that would be one way to go, but you're shutting down the whole rest of the process if your acetyl-CoA enzymes aren't able to break it, aren't able to change pyruvate into acetyl-CoA. Okay, and this is, now we just skipped the Krebs cycle, thank goodness. And now we are in the electron transport chain, and you need to orient yourself because I have seen these be in all different directions. So you got to figure out what the heck is going on. So the first question is this right here inside the matrix or between the two membranes? It is. It's between the two membranes, which is not the way we draw it, right? Usually we draw it the other way because I draw it on the bottom for some reason. I should draw it on both sides, but you just have to figure out your hydrogen, your proton buildup is going to be in between the two membranes, right? And then it's going to feed back into the matrix, even though it says it on there. I should have covered those up, you cheaters. Okay. What is the purpose of the electron transport chain? Maybe this is important. I don't know. Get your proton built up again. And what happens when you build up protons in the cell respiration electron transport chain? Where do they go? Where do your protons go?
Or the Krebs cycle.
Nope, we're done with that. We're at the very end. So we built up our protons, those green things at the very top, and they're going back through what? ATP synthase. Yeah, because we're going to make a bunch of ATP, and that's kind of important, I think. I don't know. What picks up the electrons in the end? Gets rid of them. Oxygen.
Sort of last.
Final electron acceptor. Yes, good job. Oh, there he is. I just said that. There's the role of oxygen. And there he is. Oh, we're going to say the steps. Okay, blue one on the left. What's the name of that step? There's glycolysis. Makes pyruvate. Pyruvate's converted into what? Acetyl-CoA. What is the next step? Krebs cycle, and we make some things in it. Oh man, I skipped it too fast. Dang it! Hate my life. Oxidative phosphorylation, which is comprised of electron transport and chemiosmosis. Holy cow. If it asks you, where does the cell get its energy to do this? What's a good answer? It could be anything. Where do our cells, like it said on the firefly question we had, where do the fireflies get the energy to do this? The mitochondria or whatever you want to call it, cell respiration or something like that, you know? So basically our cells get our energy to do stuff from this process right here. Okay, cool. What if there's no oxygen available? Here comes our NAD plus regenerating fermentation. Oh, see, I kind of like this diagram. Maybe I should have used it before. Its whole entire purpose is to regenerate that NAD plus at the bottom. See that? Otherwise, that can't go anymore. And going from glucose to pyruvate actually makes us a couple ATP, and so we can keep going on ATP a little bit as long as we have NAD plus. And then if you want to stay for the cell cycle, here it goes. There it is. It's exciting. What's the stopper? What stops the cell cycle?
Cyclin dependent.
That doesn't stop it. What stops it?
Those little things.
It starts with a P.
P53.
P53. P53 is our stop sign. And what is the go?
Cyclin.
CDK, cyclin dependent kinase. So P53 regulates CDK. Okay, so you just need to make sure you know the stop and the go. If you know the stop and the go, they'll give you the rest of it. What is the inhibitor? There it is. Happening, blah, blah, blah. Okay, and then there's our replication. Do you guys know replication okay? Anybody have any questions about any of the enzymes or anything? Abby's going to flip a desk if I give one more topic up here.
That's for cell replication.
Ooh. Leading strands versus lagging strand. Give me two leading strands. Wait, look at them, and I want the A, B, C, or D.
A and D.
A and D, there you go. A and D, because they're moving towards the replication fork in one continuous strand, right? See the fork movement signs? The one's going to the right and the one's going to the left.
Wait, so A and D are leading?
Uh-huh.
Okay.
Because they're making one continuous strand towards the movement. Remember what I said to do if you get this? Remember I said, like, grab your little Scantron and cover up half of it and be like, okay, which one's the leading strand? If my fork is going that way, this one is. And then flip the side and be, what's that one? It's that one. So it's A and D. Now I wonder where I got these papers. It's gonna be so, oh no, now I'm throwing them on the floor. Never gonna find all my papers I put in the grade book. I need a grade book fairy. Okay. What is this? Oh, that's mitosis. Oh, hey, I found a box plot. I put it on here for you. So cute. I think you can read those. Can you read those? You're looking at quartiles. So which quartile is Oklahoma in for the cumulative incident rate per 100,000 persons of cases? I don't know what this is. Cases, yeah. That'll be the one on the left. We're in quartile 1, right? Which quartile are we in and for the deaths? 2. Well, that was in the beginning. We got worse than that. Okay, cool. I'm not gonna, all the rest is just little data charts and things. I didn't go over ecology and evolution. Those are the 2 easy ones. And plus they weren't on the test last year. So I didn't have them on my little review. All right, if you want to hang out, look through your mock, ask questions, you're certainly welcome to.